What's going on, everyone? Here is a relatively new paper about aging clocks and the aging gaps that you get from them and how they have a lot of biological significance. So this came out in 2024 in August. I've seen it getting cited in quite a few places. There's been a new video from this YouTube channel where they talked about it. This is one of my favorite channels about aging and all the current research about it. So this comes from, they use data from the UKB Biobank. UKB Biobank has the largest data set of individuals with data to do this type of analysis for. So they used the Olink platform. This is simply a platform that measures protein levels. There's traditionally 5,000 proteins whose levels can be measured with it, but they condense it down to 2,896. That's because there's a lot of individuals with missing protein values and stuff like that. So some background: if you don't know what an aging clock is, it's basically just a machine learning model. It can be anything you want. Lasso regression is used a lot. Random forest, different deep learning models can be used, where the Y variable, the response, is chronological age. So just your age as a float value from when you were born. So historically, these clocks started with DNA methylation data being the predictors. So they're just coding 0 or 1 for whether you have a certain CpG methylation at a particular DNA site. And these are the Hannum and Horvath clocks that you may know about. There's transcriptomics, metabolomics. This study used proteins, proteomics. There's a platform that uses a lot more protein, 7,000, but there just aren't many biobanks that have all the data necessary to do this type of analysis. And you're going to see the theme that this is what is limiting a lot of research progress, is just the fact that we don't have enough data. You can also build these clocks using other things like glycomics or microbiomes, and things that are even less common like brainwaves. This is something I've done in a previous project. We actually used these headbands, got data from 700 people that used headbands during their sleep, extracted features, and in the end, we got a model that predicted age within 7 years accuracy. So it's pretty good, and I think this is going to be huge in the near future. All right, so going back to the study, they used these protein levels to build an aging clock, and they got a R value of 0.94. So it's pretty good at predicting your age based off of just your protein levels. From that, they did some feature engineering. I believe they did Baruda, a method called Baruda, which extracts just the features that are the most predictive of your response variable. And they got 204 proteins that were most associated with age. And then they validated it. This is the unique thing about this study, is that they validated it on other biobanks, people from China, which had 4,000 individuals, and also Finland biobank with almost 2,000 individuals. And this is important because it showed that these clocks made out of protein data are generalizable to other ethnicities. So it's not just this one model that's fit on a certain group of people. It has a lot of predictive power on other ethnicities as well, which is pretty cool. All right, so here's an overview on the study design. We have the data set I mentioned, the UK individuals, 45,000. You take 70% of them, build your training sets out of them, train your model, and then you can see how well it predicts age by plotting age versus the predicted age. You see, it follows along the line, which is a good sign. It shows that it is predictive, hence the large R value. Then they independently validated it, or externally validated it in this China data set, Finland data set. and tested the accuracy using those 204 proteins that were extracted using Baruda feature extraction. And then what they did, this is the interesting part of this study, is they took the residual of these observations. So residual is just observed minus predicted value. So your observed age minus your predicted age that the model gives you. They call it prot-age gap. And this is a measure of how healthy you are. So if you lie above the curve, that means that you tend to be less healthy because your actual age is higher than your predicted age. And then if you fall below the curve, so I guess in this case, they're actually plotting it the other way. So if you have a higher prot-age gap, you're going to be healthier than if you have a lower prot-age gap. And then they did association tests of this prot-age gap with different measures of your health to see if there's any statistically significant results. So a running theme of this study is the concept of generalizability. So they show generalizability in one way by seeing whether the model is still predictive on the Chinese and Finland data sets. And another thing they did to verify generalizability is look at the same individuals across different visits. So this data set from the UK, there's duplicate values. And what this means is that there's people that had their proteins measured in 2006, then in a second visit in 2014, and a final visit in 2019. And what you can do is build a separate model on the individuals from a specific time group. So it's the same individuals in each model, but it's their measurements from a different visit. And then they build the model, get their beta coefficients. If the beta coefficients are similar across each model, it shows that the model, via these proteomic levels, is picking up some kind of signal that's a function of your protein levels, right? So it's generalizable across different age ranges. There's certain rules that it's learning just based off of proteomics. And that speaks to the generalizability of this protein clock. Also, they did some further feature engineering. So they had the 204 proteins that were created using Baruda feature extraction. They then identified 20 proteins that achieved 95% of the age prediction performance of the 204 protein model. So this is interesting because these are 20 proteins that explain a huge chunk of the variation in the response variable. And if you look at these 20 proteins, these could be things that you can learn about what exactly is, most predictive of your rate of aging, which types of proteins or cellular processes contribute to your age the most. And that's what they did in this study a little bit later on. And the next thing they did was actually analyze how your aging gap is associated with different health outcomes. And they found that increasing prot-age gap was associated with increasing levels of kidney biomarkers, cystatin C and creatinine. So I don't know what cystatin C is. But these are traditionally biomarkers that are used to assess your health. Different liver enzymes, C-reactive protein, and also, interestingly, telomere length. And we know, if your telomeres are longer, you tend to be younger because there's been less cell divisions, and it sort of conserves the telomere length. And we see that the association here is significant. So there's a significant association between your telomere length and how big your aging gap is, which is some really interesting information that, based off of just your protein levels, you can tell how long your telomeres are. They also associated prot-age gap with some self-reported health variables like just self-rated health, slow walking pace, sleeping 10 hours plus per day, having frequent insomnia, and so on. And these were all significant here. And the blue versus red represents the model that was built using 204 proteins, and the one built using the 20 proteins that were the most predictive of age. And as expected, they tend to co-occur together, so they're have the same intervals generally. They also looked at the association with common diseases, and the results were, as you expect, people in the top 10% of proteage gap, so these are considered like the less healthy people, a higher proportion of them by far tend to have these different diseases such as all-cause mortality, stroke, macular degeneration, diabetes. Everything on this chart, they tend to do worse on. A higher proportion of them, if they have a worse predicted, if they have a worse aging gap, they tend to have these bad health bad health outcomes as well. So the last major part of the study is they analyzed how these most predictive proteins of age, the 204 APs, aging proteins, and the 20 APs, which proteins actually go into them, right? So first they did a network analysis of the 204 proteins using something called the string database. And they did this to identify hubs of activity that these proteins are associated with. And they found proteins like this one involved in cancer drug resistance, platelet count, this one in certain immune system functions, this one also in the immune system. This one is involved in something like handedness, so whether you're right or left-handed, which is kind of interesting. But there's a lot of different categories that these are involved in. And I think that's not surprising because aging is such a complicated process with everything's involved in aging, right? So this should not be a surprise. So these are they did another method to see what is significant. These interaction values, this is from the model itself. When you get your model output, there's also this SHAP interaction value that gets spitted out. These are some other proteins that were found to be significant. And then similarly for proteage gap 20, these 20 proteins, there were a wide variety of functions: cell adhesion, extracellular matrix, immune system again, neuron structure, development, and differentiation. Pretty much everything under the sun. All right, so on to more of the discussion. They compared the set that was their proteins with these DNA methylation clocks. And then as I showed earlier, these are just clocks built off of methylation sites. So they're like the most famous and oldest types of aging clocks. So the genes that correspond to those methylation sites didn't seem to line up with the actual proteins that are a result of those genes coding mRNA and then going into a protein. And they said that these DNA methylation and proteomic clocks seem to converge on different gene sets. So they're different. They pick up different patterns, different rules based off of their data. And then also they compare this to a previous study that also built a protein aging clock. And they found that their top 20 proteins didn't correspond to the top proteins from that proteomic clock. And that's because they said it here, it's the other study, they used SomaScan, and they used from the UKB Biobank Olink. And so there's differences between these. Some proteins are picked up less often, some are picked up more just through the nature of the technology. And that's why you have those differences. But it's still interesting that we're getting, both studies got like high predictive performance at age. So more than one way to skin a cat, essentially. There's so many rules we can learn from these aging clocks. And this is just the start. Like, this study is from my knowledge, the only one that actually looked at the data from UKB Biobank, the largest database of biological data you can build these clocks from. And I learned that they're actually coming out with more data. So this one's 50,000 individuals. In a couple of years, by 2027, according to them, there's going to be 500,000 individuals with way more proteins as well. And we're going to learn a lot more rules on how aging progresses based off of proteins. Please let me know if you like this, any advice. Also linked my newsletter in the description box. You can check it out there, along with some papers if you're interested in this and you want to read more about it. Thank you guys. Peace.